have opened already many.
Oh, man.
Of the
This is an extremely interesting field.
The funny thing is really what you so nicely, play virtuously, is the world of vitamin D. Is what I actually want to convey to the audiences. The world of vitamin D is very colorful.
Yes.
It's a wide range of options that vitamin D, where vitamin D is involved in a positive way, but to use it, to get it there, we have multiple mediator proteins.
Yes,
But they are all also from this D-binding protein kind of family.
yes,
So the D-binding protein is actually the one that makes a vitamin D, or that gives the character to the vitamin D. Which kind of character they
distributes it. It distributes it to the different tissues.
Yes.
And what we found is this vitamin D binding protein has several sugar rests connected to the protein on the surface.
That was my question. How big is the D-binding protein?
The D-binding protein is about 58 kilodalton.
So it's quite a big.
Yeah.
protein.
Yeah.
Is it?
It's not the smallest one, but
kilodalton
it is.
1,000 U, or is it?
No, they are.
I don't know, because I always have in my mind the how's it called, hydroxyethyl starch as a plasma expander, which to be a very large molecule, and that is so large, I think 500,000 or so, that it gets stuck. Also, it cannot.
So 58 is quite. It is
so nicely.
58 is quite a smaller one.
And
It's not the big. It is not the biggest. It's not a huge protein.
it is hence smaller than one that doesn't pass the kidney. Because I think the plasma expander, the effect is it stays in the bloodstream until it's somehow metabolized and attracts water, and therefore you have a bigger volume.
Yeah.
And the function is that it cannot pass through the kidney.
So D-binding protein is mainly the albumin.
Yeah.
Yeah, it is part of the albumin fraction.
So it's a medium-sized molecule.
Yeah, that's right. Yeah.
Okay. So, and very complicated with sugars, as you just.
Yeah, yes. You see, to explain what years ago they were all laughing at me when I started with explore something with glycoproteins. So glycoproteins are now more and more recognized because they have special functions. On blood group B, we have lactose on the surface with the milk sugar, lactose.
Yeah.
On blood group A, we have N-acetylgalactosamine, which is a little bit modified form of the lactose. On blood group AB, we have both. And on blood group 0, nothing. And the reason why you can take blood group 0 and put it in any other person and can make a transfusion is you don't have the sugar on the surface, which is recognized by the immune cells and attacked if it's not the right one. So what has been
Bloody sugar.
the bloody sugar, it is killing you if you take the wrong blood group and make a transfusion with the wrong sugar on the surface of the erythrocytes.
And the lactose is not the problem.
Yes, it is. It is a marker.
Also, sugar.
No, the sugar, the lactose, or the N-acetylgalactosamine
All of them.
is the marker for the immune system. You belong to me or you're foreign. And this and just to demonstrate how important these sugars are. Now, what we did is we took this D-binding protein or something else to activate this vitamin D for immune cells. You have to have a special condition. So there is an inflammation situation. You have T cells, you have B cells, and you have macrophages. So the macrophage is the one who is the conductor of the whole orchestra. And if the debinding protein is swimming along activated B cell, one sugar is split off, the lactose. If it swims along an activated T cell, the second sugar is split off. And then you only have one sugar left. And the whole protein changes in conformation. It folds, turns, flips, and the vitamin D, which has the binding area normally here, the vitamin D binding area goes into the center to be most prominent. And then we have the form which is engulfed into macrophages. And with this, you start the game of the orchestra.
Okay, good. Now, there were lots of difficult passages in your explanation.
Good.
Number one, so you have this debinding protein with the vitamin D, and it's swimming in the bloodstream.
Yeah.
And it contains lactose and galactosamine.
N-acetylgalactosamine, lactose, and
a tail of rice and other things.
Another sugar which I'm not going to explain because it's too complicated.
And basically what happens with a protein of this size, of the medium size, and with all these endings, is that it can fold and it can change its three-dimensional shape.
It's shaped.
Yeah. So now it swims along, and that you were mentioning an activated B cell. What is an activated B cell?
So a B cell which is alarmed and is producing
It's a white blood cell.
Is a white blood cell. T cells are white blood cells. They are all immune cells.
I thought the T cells are the ones that are producing antibodies. Is this correct?
B cells are producing the antibodies. The T cells are the attacking.
Yeah.
Either
And
cytotoxic.
which one is producing
The B cells.
histamine?
The T cells.
T cells. They are producing the T.
Yes.
Which one are the mast cells?
Mast cells are a kind of, macrophages, which can go through the body like amoeba.
So you think the macrophages, or you not only think, you know, macrophages, who are, as we said in the previous period, your pets at home, but
My own pets.
the macrophages, they are the conductors of the orchestra. So they somehow also control or give signals to the B cell, to the D cell.
For example, they go to a bacteria, engulf the bacteria like an amoeba, then split the bacteria into several parts, not only to deactivate them. Then the most important part is presented on the surface for the T cell. And if the T cell comes and connects to a receptor plus the new bacteria fragment, then it is activated, and then the T cell knows what to defend, what to attack.
But it's not the antibody that it makes. Okay.
Well, how are you going to produce an antibody for something you don't know for? So it will never fit. So you have to know what antibody you have to produce, where it should fit. And the macrophage is demonstrating this is the region you have to attack. And then the antibodies are produced for this part.
Macrophage. I would say, come, I have here something, yeah, and I have presented on my surface. Then my helpers, the T cells, and also the B cells.
Yeah.
Everybody is coming basically and reading what I have on the surface.
Or the macrophage is going back into the spleen and is conditioning the T cells and the B cells within the spleen.
Maybe
It's another the other option.
Yeah, those are the options. So I carry that anyway, as long as I live or as I'm there.
Yeah,
I carry.
this is how the system is working.
And that leads me also, either we could go from one to the other, because I remember also, or I know that you also found a way to analyze things that the body went through or has on the surface of the macrophage, because you can read the macrophage actually inside. It gives you all the information actually about the things that we are searching in the body, but that is for another topic. Now we are still trying to understand what happens with the vitamin D. So the vitamin D, with this nice medium-sized protein, is passing our activated T cell.
Yes.
Now the activated T cell, what does it do?
It activated
sugar. It removes the
Yeah, well, has a galactosidase on the surface. If it is activated, it produces galactosidase on the surface, and then it splits off the galactose on the surface of the debinding protein.
So again, the chain of causes is, so the macrophage sees, let's say, a thread, engulfs the thread, puts a remarkable part of the thread out on the surface,
Yeah.
activates the T cell is activated. Now, then a vitamin D with a
Then the vitamin D, with the activated cells, the vitamin D is prepared to go to the next macrophage and put him into alarm.
Yeah.
So this is the form how our immune system is activated by vitamin D.
listing basically
Yes, and
protein, the
this vitamin D is going into the macrophage, and this is what we have investigated. It is activating about 2,100 different genes, and from the genes you have a production of different proteins, markers, and alarm substances. So this starts the whole process.
Now I want to know, did we discuss enough to put butter by the fish? Yeah. I would like to now say I understand. There is one macrophage, one lonely macrophage, and it comes across, let's say, a cancer cell, and it recognizes this cancer cell, and it eats the cancer cell, and it puts the remarkable parts of the cancer cell out there.
Yeah.
And it happens also to pass a T cell, and the T cell gets activated for this specific remarkable part of the cancer cell.
Mm-hmm.
Now, the vitamin D with the right protein passes this T cell.
You're on a good way.
And the T cell removes certain or changes the protein in a certain way. And now the vitamin D is able to ignite a fire in another macrophage.
Macrophages are like guardians sitting in any of our organs and everywhere.
And these macrophages, and what will they do now? If the vitamin D goes now in the macrophage,
Then they are attacking the next. Then these macrophages are attacking the next cancer cell and are activating the yes. Then they're whole
putting this on.
Then the whole cascade starts.
Yes.
But
you need to know the right protein.
You need to know the right protein. You have to have an activated macrophage, and the tumor cells are many tumor cells are able to produce a protein which is called nagalase, which splits off the last sugar from the debinding protein. And then this is inactivated. So the cancer cell has a possibility to deactivate this whole system. And what we frequently see is you have many immune cells within a cancer region, so-called tumor infiltrating lymphocytes, who are not active. And the question always was, why are these cells not active? Because the tumor cells are able to calm down the immune cells. And therefore you don't have an activated T cell, you don't have an activated B cell, and then you cannot convert vitamin D, and then the cancer cannot be offended.
And is there a solution?
Yeah, I will. We went and remodeled this vitamin D binding protein. We bound it to vitamin D, split off the sugars, and now you have the starter. So we produce the substance you need for ignition of the whole process.
So you have already
decided about
the twisted protein.
Yeah, it is a ready twisted protein containing the vitamin D, fitting to macrophages and T cells to activate them, and this is a more or less harmless liquid.
It's like a sport or like a football coach who says, yalla, go, yeah, and bends them all up.
Yeah, this is like vitamin D is the fluids. Is the gas that you use in a motor. But you cannot start the system without ignition.
Yes.
And the immuno D is the ready preformed combination protein in right shape with vitamin D to make the ignition. So
That's amazing.
there's two parts. You need a good vitamin D level. That's right. But if you just have gas in the car, it will not work. You have to